Okay, welcome to Ask the Expert. Today, we are very pleased to have Dr. Eddie James speaking to us about applying in-depth studies of antigen-specific T cells to understand disease progression and response to therapy. He's coming to us from the BRI in Seattle. And just a little bit about him, he earned his PhD in biochemical engineering at University of Washington. After an internship at Zymogenetics, he joined the BRI, or the Benaroya Research Institute, to manage their Tetramer core. And he currently is leading several projects related to the role of epitope-specific T cells in autoimmune diseases. Particular interests of his include studying how protein modifications and beta cell stress contribute to the breakdown of tolerance, and applying in-depth phenotype analysis to investigate changes that occur during autoimmune disease progression and in response to immune therapies. He's a member and co-leader of a working group aimed at improving disease staging. This is a critical path, really. Disease staging of T1D patients through the use of validated biomarkers, and participates in multiple collaborative projects which characterize T cells in autoimmune disease using at-risk individuals and clinical trial participants. This is a real strength of the BRI up there in Seattle. And he also is very involved in performing sensitive and complex immunological assays using blood samples from variable sources. So that's, he's really directly on the human immunology front line. Thank you so much for joining us, Eddie. I really appreciate it. And we have a really interesting group in our audience, so I can't wait to hear what you have to share with us.
Well, thanks for the introduction and for the invitation to present. It is a privilege. You guys see my title, and I guess do people usually ask questions as we go, or do we save those for the end?
Whatever you prefer.
Okay. Well, I guess if you have a burning question, feel free to raise your hand, or however we do that.
Yeah, you can raise your hand, and
Yeah.
I'll get it. Otherwise, we'll get them at the end. Sounds perfect.
Yeah. And so some of the content is a little on the technical side, but I've tried to make it accessible just in case we have, a mix of people.
Nice.
But
okay.
If anything isn't clear, feel free to stop me.
Sounds good.
So I'll begin just with a little slide to kind of, this is kind of an homage to what the mission of our lab, and that is we're really trying to break the cycle, that leads to type 1 diabetes. And the way we think of the disease is there's some trigger that's still unknown. Some people suspect could be viruses, could be diets, but in any case, something kicks off an autoimmune response against the beta cells in the pancreas. They get selectively attacked and destroyed. But we see this as a multi-wave process where basically you have an initial attack. This leads to dead and impaired beta cells. This frees more antigens, which get brought around to the lymph nodes, engaging a second wave of T cells. And basically this circle amplifies and ramps up until enough beta cells are destroyed that you get diagnosed disease. And we see this as a circle where there's nexus points that can be treated as well. And so my lab is collaborating with several others to work on things like engineered Tregs to help restore the initial break intolerance, and looking at various inhibitors that would halt the generation of and release of new epitopes, and then immune modulators that help shut down the ramping up of T cell responses that really spins us around this loop. And so we try to be really active partners. I always like to mention that this is personal for me. I have 2 nieces with type 1 diabetes. And so fighting this disease is not just an academic exercise for me. It's something that really matters to me at a personal level as well. And I think that affects how I do my science.
Yeah, I think that does. And I think your commitment to it, and we here at Sugar Science completely understand that and are all in as well.
Great. And so what some people don't appreciate, but maybe this is not news to this audience, but the diagnosis of type 1 diabetes, marked by this arrow here, it represents a breaking point where there has been enough decline in beta cell mass and beta cell function that insulin production is no longer sufficient to control blood sugar levels. But the process doesn't halt at diagnosis. The beta cells insulin production isn't zero at diagnosis, very often, unless you have an extremely fulminant form of the disease, and beta cell function continues to decline after onset, and it can be years or even decades later that someone really zeroes out in their ability to produce insulin. This is measured in the blood by a test called the C-Peptide measurement. C-Peptide it's the inert peptide is cleaved out of insulin as it's processed from proinsulin to insulin, And so that's our best way of measuring whether there is residual beta cell function or not. An important point in today's talk also is that subjects with type 1 diabetes tend to fall into two broad categories, and that's shown by this little line plot here that my arrow is hovering over now, where there's this blue line and this red line, and you can see the blue line and red line are groups, and in between are the spaghetti plots that are actually real people. So people defy groupings, but in concept, there's people that at diagnosis seem to rapidly lose their residual beta cell function, and there's people at diagnosis that tend to maintain it for quite some time before it finally tapers off. I mentioned this, and we think this is an important thing to understand because it turns out that this matters.
Yeah, and that also just sort of goes right along with the whole story of endotypes, right? That there's some very well characterized endotypes that are emerging.
Exactly, and there is, as is shown by this review article here, that there if you're diagnosed at a young age, you tend to fall more into what I'll call this red group, this low C-peptide group. Whereas if you're diagnosed at older ages, you tend to fall more into this green area, this what I call the high C-peptide group. But it's not just binary. You can see over here, there are young subjects that are still in the green, and there are older diagnosed subjects that are still in this red. But if you're diagnosed above this threshold value of 0.2 nanomoles per liter of C-peptide, it's been well documented by multiple studies now that you have fewer complications, you have lower hemoglobin A1C, you have less risk of hypoglycemia. Basically, everything goes better for you if you're in that group. And it's partially because of that for many years, the gold standard for intervention trials in type 1 diabetes is preservation of C-peptide. That's what the FDA considers this to be a viable endpoint. And that's because it actually matters in the life of the patient. We can't necessarily rewind time and give you all of your beta cell function back, although groups are working on that, and that would make a cool, talk for later. Or maybe you've already hit on that in previous talks. But preserving what you already have or preserving new beta cells that you might engraft is really important. And so the way my lab tackles this is we are experts at using a reagent called an HLA Tetramer. And what HLA Tetramers are, these are reagents that they use the natural HLA molecules in the immune system, which APCs use to present peptides to T cells. We make them in recombinant forms, and we cluster them around a fluorescent stripped avidin core. The reason we use tetramers is because it turns out that a monomer or a dimer doesn't have a stable enough interaction to stick for a long time. But if you tetramerize these recombinant HLA molecules, they'll actually stick to a T cell for 30, 60, 90 minutes, which is long enough to take it upstairs and do a flow cytometry experiment. Through a broad series of efforts, some of which I have reviewed in an article that came out a few days ago, people have painstakingly defined which fragments of beta cell proteins are recognized by the immune system. And if we now pair that with tetramer technology, what we can do is we can take an HLA molecule that matches the patient of interest. We can load it up with a peptide that we know the immune system sees, and we can use that to specifically label just T cells that are beta cell reactive. And we think this is important because when a person has an autoimmune disease, 99.999 plus of their T cells appear by every measure that we can do to be more or less normal. It's that minor fraction of autoreactive T cells that is doing something wrong. Or as you'll see later in this talk, it's a fraction of that fraction that's doing something wrong. But to see the individual trees rather than the forest, we need to be able to label the antigen-specific T cells and ask questions about them. And that's what my lab does. And so what we have done over the 20 years that I've been at BRI is we've done from doing very simple flow cytometry experiments. When I first joined BRI, we could do 4 colors at a time. We now have these very sophisticated flow cytometers where you can do even 30 different measurements at one time. And so that's this box here, that's high dimensional flow cytometry. And so if one of those colors, we can label beta cell reactive T cells. If those other colors are different immune receptors of interest, we can bin those T cells into various functional phenotypes based on known surface markers and ask a lot of questions about them. But excitingly, we have more recently paired that with gene expression profiling through a technique called RNA-seq. And for those who are laymen, I'd like to say, I think of RNA-seq as kind of like when you hit Control-Delete on your computer, and you can open up that second tab that shows every process that your computer has running. That's what RNA-seq basically does. Is it allows you to crack the cell and ask, you get a list of all the transcriptional programs that cell is running at one time. And so when we combine that with the specificity of flow cytometry and HLA Tetramers, we can, in-depth and important information about, what's going on in the T cell. And finally, we compare that with TCR sequencing. And again, the T cell receptor is, to a first approximation, unique to each T cell, and it determines, which peptide that T cell sees and with which affinity, what activates it. And so by doing TCR clonotyping, we can basically, barcode the individual cells based on their unique identity and specificity. And so if we can complete this triangle, we can ask very sophisticated and important questions, high resolution questions about the immune system and how it's behaving. And so our goal then is to apply these techniques, generally as a lab to important sample sets. And what I'm going to talk to you about today are studies that we're doing aimed at understanding this phenomenon of C-peptide loss in subjects with established type 1 diabetes. And so our goal for this project, which we hope will be published soon, is doing single cell RNA sequencing, in combination with HLA Tetramers and T cell receptor analysis on samples from subjects with established type 1 diabetes who at the time of sampling had either high or low levels of residual C-peptide. And so we're doing tetramer analysis by flow, TCR sequencing, single cell RNA-seq to really characterize, an immune fingerprint in these cells that associates with having C-peptide loss or C-peptide maintenance, because we think understanding that is important. And
that's a great approach. I wonder how many subjects or samples are you looking at in that context?
So I'm going to get into that in the next couple of slides, but to give you a quick question. So, for the in-depth analysis, we've done 10 subjects per group. Mm-hmm. For the broader, just basic flow cytometry, we're able to do much larger groups. The reason is the single cell analysis is still very expensive.
Yeah.
We're streamlining it, but especially at the time that we began this work, doing it in a lot of cells, it was cost prohibitive, and we estimated that a group size of 10 would give us enough initial insights to justify this project and to motivate further work and further funding. And so that's where we landed for this. And so this is that study cohort. And so here we have 10 rapid progressors, 10 slow progressors. And again, we're using that threshold level of 0.017, which rounds up to 0.02 nanomoles per liter of C-peptide. And to define people as rapid progressors, we wanted them to have lost measurable C-peptide, being below this measurable threshold within a relatively close time to diagnosis. And so all these subjects had an average of 2.8 years from diagnosis, and they had already lost basically all their measurable C-peptide. So kind of an extreme group. For slow progressors, we didn't want to call people slow progressors if they were about to lose all their C-peptide. And so we wanted people to be, well above that detectable threshold. So these subjects were anywhere from 0.14 to 0.58. And these subjects were all at least 5 years post-diagnosis. And so these are subjects who had maintained C-peptide and they've been maintaining it for a while. So again, another extreme phenotype. And so our hope was that by comparing these subjects that we gain some important insights about how antigen-specific T cell responses might be different in subjects that rapidly lose their C-peptide versus those that are able to keep it for a while. Because again, these are subjects that tend to do really badly and have very brittle diabetes, like my older niece, or who do better and have an easy time with glycemic control, like my younger niece. And so we began with just some very basic assays, unsophisticated assays, just to make sure that there weren't just simple, easy to detect differences between these two groups. And so we took a well-described panel of HLA class 1 tetramers shown here. We didn't develop this panel. This was actually developed by Bart Rupp's group, well over a decade now. And these are 6 well-defined class 1 epitopes from what we consider to be kind of the primary autoantigens in type 1 diabetes. And so we have the best epitope from GAD, IGRP, IA2, preproinsulin, one from the mature insulin B chain, and another from the zinc transporter 8. And these also correspond to the major autoantibody specificities. These are well-behaved tetramers that have been shown to stay in reproducibility in subjects that have type 1 diabetes. And so we can visualize the fairly rare T cells for each of these specificities, and we can either count them individually by flow, or we can pool them together and advance them for single cell analysis. And first, we wanted to ask just a very dumb question, and that is is the only difference between our high and low C-peptide groups how many T cells that they have? And as is shown in this little scatter plot down here, there's a slight trend towards people with rapid progression having a few more antigen-specific T cells. This is in cells per 100,000. So these are pretty rare cells, but there's a lot of overlap between these groups. And so it isn't a case where, if you have very few T cells, you do better. And if you have a lot more T cells, you do worse. It's just, if you have rapid loss of C-peptide, you have a little bit more, of a high number of T cells, but it's not super convincing. We also wanted to ask about, kind of the distribution of the specificities. And so we binned the T cells amongst the 6 different specificities and just kind of asked, is there some, evil epitope that's more frequent than all the others? And, comparing our rapid group to our slow group, and expressing the average frequencies as a pie chart, there was a surprising conservation where the hierarchy of these T cells tended to be pretty consistent. Where IGRP tended to be the lowest, GAD tended to be the highest, and there really wasn't one epitope that stood out as being something that might be driving rapid loss or slow loss of C-peptide. We even asked whether there was just more generalized diversity between the cells by saying if a subject's T cells are 20 marbles, do they tend to be spread out amongst all 6 of these bins, or are they grouped within just 2 or 3 of these bins? And there's some sophisticated diversity measures you can calculate for that. And again, our rapid group tended to have a slightly higher diversity index than our slow group, but it wasn't super convincing. And so that reassured us that to really understand what was going on with the T cells, it's not just a numbers game. We have to look at the character of these cells, and we have to look at their transcriptional programs. And so that's exactly what we did. And so we turned the crank on our single cell RNA sequencing pipeline, where basically, again, we stain cells with Tetramer, we stain cells with a panel of well-described surface markers to aid us in classifying those cells. And basically, we sorted single cells onto plates using an in-house system where we could simultaneously, entrap these cells and send them for RNA-seq analysis. We can assemble their T cell receptors, and we can capture their surface marker expression for all of ours of interest, and we can cross-index all 3 of those data types and ask questions about what differentiates our slow and rapid groups. And to make a long story a little bit shorter, what we found, is that what was the most interesting in these data sets was actually their overall single cell RNA sequencing profiles. And I don't know how acquainted this audience is with single cell RNA sequencing data, but there's thousands of parameters that you get out of the single cell RNA data set, and that's something it's difficult to wrap a mind around. It's this huge matrix of values. But there's these projections called UMAP that basically it takes this highly dimensionalized data and it projects it onto a planar surface so that where each spec is an individual cell, and it uses these UMAP parameters to help define relationships between these cells, such that if 2 cells are close to each other on these UMAP projections, they have highly similar properties. If they're far apart from one another on these UMAP projections, they have very different properties. And then the program, it then defines the reasons on this UMAP as clusters, where clusters are basically T cells that are near enough neighbors to one another that they share phenotypic traits. And so that's exactly what our analysis platform does. And so it took, the thousands of cells that we obtained by single sorting cells from these higher C-peptide low subjects, and it sorted them into this UMAP landscape of T cells, and it clustered them. And what really came out of this is that there's these distinct phenotypic clusters of cells, and of course the next task then is to figure out, okay, what do these clusters mean? And so the way we really grapple with that is doing things like overlaying what we learned from the protein expression onto these cells. And a good marker that we like to look at are things like CCR7 and CD45RA, because these are markers that allow us to tell how antigen experienced a cell is. And so that enables us to kind of draw a gradient on our cell from more differentiated to less differentiated T cells. And looking at CD45RA and CCR7 expression, what became clear to us is that this upper point here where I'm hovering my mouse now, can you guys see my mouse okay?
No, I don't see it.
Okay, why don't I try to switch to, because you're gonna have a hard time following this.
I do see it. It's sort of moving around. Yeah, there. That's good.
So let me try to switch it to like a spotlight. Is that well? Yeah, I'm used to a different program than can you see the red dot now?
Yes, that's
so hopefully that will stay. So this upper corner here, is the least differentiated area, and this lower quadrant here is the most highly differentiated area. And what stood out to us as interesting is that, you know, if you look at this like a kind of a developmental stream, what we can see is that, you know, from the most naive state, there's this upper path and there's this lower path of differentiation. And that was very interesting to us. We looked a little bit further at our markers. I'll advance here to the next slide. And you know, one thing that we looked at was, you know, the markers KLRG1 and TIGIT are exhaustion markers that tell you that a T cell has basically become burned out and become inert. And previous studies have shown that if exhausted T cells seem to be protective, you know, in autoimmune diseases. And what we saw then is that, you know, there definitely is kind of, you know, this upper branch and then this lower pool here seems to be kind of a path towards exhaustion. Whereas the lower branch in the central region seems to be a more non-exhausted path. And so there seems to be this fate where a T cell starts, you know, at this point being non-differentiated, and it can be driven along one or two paths. It can be driven along a path to become a fairly inert, exhausted cell, or it can be driven along a path where it can come to rest as more of an effector cell, or there seems to be this reservoir, you know, in between the two paths. And so those are phenomena that we wanted to zero in on. And so this kind of shows that you know, that same cartoon that we seem to have this exhausted path and this effector path. And this is held up by other markers. And so if you look, you know, at things like granzymes, you know, and chemokine receptors that seem to illustrate more of a, you know, effector-like cell. If we look at, you know, molecules like LAG3 or EOMs that seem to signal more of an exhausted signature. There definitely is, you know, basically two different fates where these cells can come to rest, you know, exhibited by different clusters. And so, of course, the next obvious question is, okay, we have these different cell states, anywhere from naive to exhausted or effector cells, or this weird reservoir state. What differentiates our high and low C-peptide group? And what we saw then is that, you know, there's this exhaustion axis, you know, this EOMs axis, and it's hard to see in this plot. We have blocks here that represent higher C-peptide low, but I'll digest this for you in this next slide. What we saw is that, you know, if we take a look at the cluster proportions between our high and low groups, we can see that our C-peptide high group versus C-peptide low groups, they differ with respect to these three key clusters. And what this really boils down to is that, you know, our more, you know, exhausted clusters seem to be overrepresented in our C-peptide high group, like this cluster 8, whereas there's these bad clusters, this cluster 7 and cluster 1, that seem to be overrepresented in our low C-peptide group. And so this really shows that there really are these good guy cells and these bad guy cells that seem to differentiate C-peptide high from C-peptide low, which I thought was pretty fascinating.
That is.
And so we can also recapitulate this by using a previously published module called this EOMs module. And again, in our slow progressors that maintain their C-peptide, we have this very high level of this EOMs exhaustion module, whereas we have a lower, you know, expression of that module for our rapid progressors. And so this story really seems to be holding up, that there's these two developmental fates, you know, for autoreactive T cells. And if your T cells get pushed in the wrong direction, you do worse. We also wanted to ask about T cell receptors. And again, if you recall, you know, a few slides back when we made our hypothesis about this test, we really thought that one component of this was going to be that T cells that were more driven, you know, might change your fate. And so we were surprised to see that if we looked at, you know, diversity or you know, clonal expansion in these T cells, which is shown by this circles plot, whenever T cells are sharing a T cell receptor when they're sister clones, then they're connected by these little arches. And so you can see these little patches of sharing. You've got this red sharing and this pink and dark blue and light blue sharing. And we saw a few interesting phenomena. Number one, we saw that there's almost no sharing between individuals because each of these colors is an individual. And so, you know, if people, they seem to have personalized individual repertoires of these cells. But we were a little surprised to see that the amount of sharing didn't really differ between our rapid and slow groups. And so the level of turnover of these cells, you know, the degree to which they've been pushed by antigen seems to be fairly similar in these studies. And that may be why when we did our more simple flow cytometry assays, why they seem to have similar numbers of these cells, which we all thought was pretty interesting. But the other thing that was, you know, the other thing that we noted was that, you know, this airline plot shows basically T cells that have the same T cell receptor, and it connects them with lines on this airline plot. And what we can see is that, not surprisingly, T cells that are expanded seem to be not in this naive part, but in this differentiated part, which makes teleological sense. But what we notice is that T cells that have the exact same T cell receptor can live in different areas. And so it's the T cell's life experience and that specificity that drives the character that it takes on. And so that means that basically, any T cell specificity is a candidate to be dysregulated and cause disease, or any T cell receptor is a candidate to be re-regulated and to be more inert or maybe even hinder disease.
A lot of plasticity. One question. Did you color your UMAP by your two groups, rapid versus slow, coming from University of Florida?
We have done that. I don't have that slide in this presentation, and but the dots do segregate.
Okay.
But it's I took that analysis up. It's difficult to see by eye because again these differences are subtle. Yeah. But yes. Okay, and so kind of where we've landed, as far as our conclusions from this study, is really that here based on the single cell analysis, what we have seen is that there seems to be two distinct developmental trajectories that the CD8 positive T cells that are autoreactive can take on, and this third kind of reservoir state that we're kind of interested in, and that increased exhausted phenotype is associated with higher residual C-peptide. We see that islet antigen reactive cells that are both in effector and exhausted clusters are more expanded and less diverse than naive cells, which isn't surprising, and that more expanded and less diverse cells were slightly associated with higher levels of C-peptide, but the differences weren't as impressive as we expected. And to us, this motivates interventional strategies designed to drive T cells to exhaustion. And so in the last few minutes, I want to talk about kind of what we're doing to work on that. And so for our ongoing work, we're getting T cells from patients that are being studied and at-risk subjects that are being treated with a drug called teplizumab. Teplizumab is an anti-CD3 antibody that's basically designed to deviate T cells to a less pathogenic phenotype. And this drug has been shown in just kind of studies of global T cells that one of its effects is that it can deviate cells to more of an exhausted phenotype. And so we're able to get a pilot set of samples on 12 T1D patients that we measured either at baseline or post-treatment with teplizumab. And this is a very preliminary study, but what's I think exciting is that we're seeing a lot of the same things, and that is, again, the shape of this UMAP is different, but when we do single cell analysis of these pre and post treatment cells from these subjects, we see some of the same fates, where we see in this case a naive T cell, and we see a trajectory that can work its way around to either an exhausted fate or to a more effector-like fate. And so again, there's two branches to this stream, and there even seems to be this side group that seems to be parked right between naive and effector, and this may be that reservoir state. So there's some agreement there. And what's interesting is that when we separate out, here we did either virus-specific cells or islet-specific cells. And again, this cluster 3 is our exhausted cluster, and light blue is pre-treatment, dark blue is post-treatment. And we see the cells being pushed by teplizumab more into this exhausted cluster. And interestingly, this cluster 4 is our most highly effector-like cluster, and we see cells being pushed by teplizumab out of that cluster. And so it seems like there is an inversion event happening here. And then our goal
certainly does. There's another question. How do you plan to control variability due to individuals' ages?
So that's difficult. The way we're trying to grapple with this right now, since our N is really low, and since when you're looking at clinical trial samples, you have to take who you can get, and that is that we're trying to basically use mathematical modeling to regress that out of our signal. But that is a challenge, and that's the biggest problem with these low N type studies. Is, I mean, again, when you're talking to TrialNet trying to get samples, 20 samples seems like a lot, but then when you sit down to actually do an in-depth analysis, 20 samples doesn't seem like much at all.
What would pave the way for getting more samples? I know obviously the T1D exchange was working in that space to try to connect people with trials, et cetera. But how could you really expand that pool of participants?
Well, I mean, to me, I think to be honest, I have found, TrialNet and some of the other groups to be, given the limited resources that we have and the large number of people that want them, I feel like they have actually been pretty generous with, and so I don't fault them at all. I think what's really needed to build up to the larger N studies that we need to completely crack these questions is, to have a project that is designed from the very beginning to include these types of in-depth readouts as part of the trial, rather than ex post facto, because the key is basically connecting enough samples and having them ready to go to do the in-depth analysis.
And what other disease state has that as a working model right now, to your mind? Any?
I mean, in my experience, the gold standard is rheumatoid arthritis.
Yeah, I would
where we're just about to publish a paper where we followed antibody positive individuals, and in RA it's an antibody called CCP, which is an cyclic cis-related protein antibody that like type 1 diabetes, it precedes diagnosis by about 10 years. And there was this robust multi-year funding that allowed collection of samples from these at-risk subjects at multiple time points over a 3-year period, and then we were able to do tetramer analysis and antibody analysis and other omics analysis on that grid of samples for a fairly large N of subjects. I think it was something like 40 subjects per group.
Is that driven? Was that an NIH-driven grant?
It was actually a private company.
Yeah, which is why we invite industry to these talks. So we'll see what happens. Okay, yeah, sorry to get off on a segue, but I do think that's a very important point, because I've heard it a few times that the samples are so precious. How can we get more of a a flow coming in to these new to be looked at in these new ways?
Yeah, it is. And I'll toggle ahead just because that was pretty much my last slide. And so, parked up here with my acknowledgements. And so I do want to give props to my formerly postdoc, now staff scientist Hai Nguyen, that did all the hands-on work as far as doing the tetramer staining and sorting out these cells, and Gabrielle Blahnik, who first implemented this class 1 panel when she joined the lab. And then, of course, our genomics core, our systems technology. This whole pipeline was done by Peter Lindsley, and Alex Hugh is the one that actually did the analysis for this project. Samples were selected by our BRI interventional immunology program, led by Kate Speak and Carly Greenbaum. And then, of course, our clinical core and my other co-investigators that have done this work. And now I can release you guys to ask more questions if we haven't hit on them all.
Yeah, no, fantastic. That's great. That was really interesting. And we gave a shout out to that new paper you talked about to our wider audience. Hopefully people will pick up on it and take a look at it. Feel free to unmute yourself or raise your hand, and I will unmute you if you have any questions here for Dr. James. Not at all. I mean, in terms of sort of this tetramer work and the work that you're doing to really kind of, dive in to specify what's happening during the prodrome, what's happening over there in Europe? Nodia is sort of, they've got the UK Biobank. I know, Tim Tree's heavily involved in the T cell world there. Are they going after this with the same, I don't want to say fervor, but in the same way as you guys are doing this in your laboratory?
The short answer would be yes. There's a bit of a lack of consensus about exactly what the best assays are to apply to these sample sets.
Yes, that's what I was trying to get to.
Yeah, and that becomes difficult. I mean, it's because there are pros and cons, to any assay. I mean, something like an ELISpot. I mean, people wouldn't have been enthusiastically doing ELISpots for the past, 25 to 30 years if they didn't tell you something important, and they do. There's some of these more novel, short-term activation assays, the so-called, CD154, CD69, or the CD25 OX40 assays that, are better at casting a broad net and giving you a kind of a larger group of autoreactive cells. But the cells that come out of them maybe aren't quite as cleanly positive as something like a tetramer. And so you know, it becomes a difficult proposition to decide, what's the best assay. And to be honest, maybe that's even the wrong question, because there's the old saying that you can't let perfection be the enemy of the good. And I think there are some degrees to which we kind of wring our hands and say, okay, we don't want to waste these samples until we have the perfect assay. Yeah, but well, you're never going to have the perfect assay. At some point, you just need to crack the vials, use the best assay that you have, be as smart as you can, and learn what you can. But
how might the bigger how might the role of bioinformatics and ultimately like AI come in to look at these heterogeneous assays in new ways? Is anybody doing that, or is that something that might be done in the future?
I think that there is room for a lot more creative analysis than what we have been done so far. And again, I'm pleased by what I've seen in the field as far, not only our bioinformatics department, but other high-level programs across the U.S. and around the world are doing an increasingly good job at getting the immunologist and the bioinformatician in the same room and more speaking the same language.
Yeah.
But at the end of the day, we use the analysis methods that are available and vetted to analyze our data sets, and then if that reveals a useful insight, then we publish it and follow it forward with more mechanistic studies, or maybe it inspires the development or testing of a drug if you're really lucky. And then, although in theory that data could be reanalyzed later or meta-analysis could happen, I think we need to do better at that. I think that follow-up analysis and meta-analysis is pretty important.
Yeah.
I see a hand up.
Yep, and here's Yang. Go ahead, unmute yourself, Yang guy.
Hi, Eddie.
Hi.
Is that, as you know, you said is a fraction of cells are autoreactive, so they're very minor, small percentage, which make it the technology really not that friendly for most of our immunologists. Is there any way you guys are thinking how to improve this, make it more friendly, reproducible for different labs?
I mean, that's something that we're working on. And again, I mean, you may be aware that when we implemented this HLA class 1 tetramer assay in our lab, one of the first things that we did is we cooperated with 5 other labs to do multi-center testing of that assay. Yeah, and the unfortunate truth was that at that time we didn't see really tight technical consistency of those measurements across the 5 labs. I can tell you that we've learned from that study, and we're about to submit a study, a class 2 tetramer validation that we did, where we actually see much tighter replicates. And so I think some of it boils down just to having reliable reagents, rigorously written SOPs, and understanding what causes variation. So I think that we're much closer now than we were 5 to 10 years ago at being able to teach other labs to do these assays really, really well. And I think an example of that would be I had a visitor in my lab from Chantal Matthews in Lute Overberg's lab, the heart of Anodi, if you will. And she basically spent the summer in my lab learning how to do tetramer analyses our way, and then she went back home and ended up doing a major chapter of her dissertation on some tetramer work. So it's becoming exportable.
Yeah,
so,
that's just an amazing shared resource when trainees can actually go to places, and learn from, I guess I say, from the masters, of how they do certain things, and then bring it back and incorporate it in their own group.
And AAI has things like travel for techniques, and so I would say that, I mean, if this is something that you feel like is essential for you to do what you want to do, send someone to my lab and I'll teach you. I don't mind. I mean, we're a community, right?
I love that. It's very good. All right, last call in the interest of time. Last call for questions. All right. keep up the amazing work up there in Seattle. Stay safe from all the smoke, and can't wait to hear or to look for that new paper that's coming out of your lab. Really appreciate all you're doing in the field.
Well, thanks, and thanks again for the opportunity to present, and to
Great.
those who attended. Thank you.
Thanks to everyone who attended.